Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with John Tipping, who's with Haley Ward, Inc. John is an aquatic entomologist, and I had a great time learning from him what exactly that means. Hearing about water quality and sampling from John, and how often that work is part of much larger projects, was really interesting. And I hope you enjoy our conversation as much as I did. Well, John, welcome to the Maine Science Podcast. It is a delight to have you. You might be our first taxonomist.
That is exactly right. By training, an aquatic insect and invertebrate taxonomist.
Fantastic. Before I let you explain what that is and what you do, perhaps you could tell us how you got into science, what sparked your interest, and how you found your way to aquatic insects.
Well, I've always been interested in nature in general. Even as a kid, growing up, gardening and doing work around the house in the yard with my dad, I was always the kid who was turning over rocks, looking for creepy crawlies. And I'm going to give a shout out to the teachers out there in the world. My favorite teachers through high school were always my biology teachers. And when I got into college, I was still maybe a little bit directionless and took a lot of different courses, but it was the biology courses that always stuck with me. And I was kind of at a dividing point as an undergraduate between organismal biology, and I was already leaning towards invertebrates at that point, or cell biology. And I ended up following the route of invertebrates, insects specifically. And then I started working at a company in Unity, Maine in the late 90s called Lodic Incorporated, an environmental consulting firm. And I worked there for 12 years. At that point, the owner retired and I bought the business. And then that eventually led me to Haley Ward, where I just started. The aquatic entomologists of the world are maybe, I don't want to call them the black sheep, but we're definitely in the minority, as you might expect. The majority of entomologists tend to go into agricultural or pest management or medical entomology. Those are probably the big fields that most people would be more familiar with. And the aquatic entomologists are a bit of a minority.
Well, before I have you explain what it is that you do that's different than the other ones, it's really interesting. I actually just spoke with someone earlier this week who I would say they align with the other two that you just mentioned. So the difference between cell biology and what you ended up are pretty different. How did you decide? I think that it's a fallacy that people have to decide when they're in college exactly what they're going to do their whole life. But you do have to narrow it down, right? How did you pick when that fork happened?
I think really what it came down to at the time was I didn't see myself as being a laboratory technician, which is where I thought I would end up as a cell biologist. I wanted more opportunities to do things outside. And so that was probably the biggest driver behind that decision. Of course, it's funny now because the work I do, I still do the macroinvertebrate taxonomy and bioassessment, but a lot of the work I do is also toxicity testing, which is laboratory work. And so for the last 5 or 10 years, I might have a week's worth of field work in a given year, and the rest of the time I'm in the laboratory. So
that could be as much an evolution of a career path as anything else.
Oh, yeah. But I felt like being confined to laboratory work, I wasn't going to enjoy that. So that's why I ruled out cell biology at that point. But I still just find it fascinating. And with the leaps and bounds that have been made in technology and genetic analysis and molecular biology, it's a fascinating field.
Yeah, it feels like that's the next big frontier. You know, we had space and computers, and biology is just almost unrecognizable, I think, from 50 years ago.
Oh, absolutely. I think fields like physics too, and similar in the way that oftentimes you'd hear quoted, there'd be a physicist 50 years ago who'd say, well, we've learned everything we can learn. And then it just never ends. And I think that's one of the great things to think about just about science in general is how it's a process. It's a learning process. It's a discovery process. There's always something else to be learned.
So fill us in. What's an aquatic entomologist do?
Well, the reason that aquatic insects are useful in a practical sense is they're indicators of environmental conditions, and in particular, water quality and habitat quality. Aquatic insects, like all other insects, of course, they undergo different life stages. And the immature stages, the larvae and the nymphs, tend to stay confined to a particular sort of habitat. And I'm talking about not just a stream or a river, but a particular subset of that. They like certain substrates. Some live in the mud, some live on the rocks, that sort of thing. And they do move around. They drift downstream with the current at night. They crawl or swim upstream during the days. So they are moving around, but they stay put. So if there is an environmental impact, they're going to notice it. And there are hundreds of species of insects that have aquatic life stages, and they all have different tolerances to different types of pollution. And it's a fairly simple procedure, if you know how to identify them, to go into a habitat like that, collect a sample of the aquatic invertebrate community, and make a determination as to whether there's an environmental impact. And so as a bioassessment tool, this is something that's been used for decades by regulatory agencies from the municipal and state level up to EPA and USGS and organizations like that. And so a state like Maine, the Maine Department of Environmental Protection, they have a model that they've been using for about 45 years now, that I can send them a data sheet with what I've collected in a wetland or a river or stream around the state of Maine, and it will give me a water quality or a water class determination based on that community. So it's a very useful tool, and it just tells us if there's an impact. If it turns out that there is an impact, then we can do more specific sorts of work. We can do chemistry analysis of the water or the substrate, and stormwater assessments, watershed assessments, to try and find out what's impacting that particular community and what we can do about it.
So when you have a water quality sample that has something that is not what would be considered normal or good, can that be indicated by either an increase in a certain kind of species or a decrease? I mean, does it go both ways, or is it always kind of like all the populations are being slammed here?
It can go both ways. And the way I like to describe it is we're not necessarily looking for particular species, but we're looking at the community structure. And so there are organisms that are very tolerant of pollution. And if you have, say, 30 species in your sample, and half of the organisms in your sample belong to a single species that's very tolerant of pollution, then you probably have an issue. On the other end of the spectrum, we talk about a group called the stoneflies. Stoneflies are not tolerant of any sort of pollution, typically. So if you have multiple species of stonefly in your sample, then you know that ecosystem's in pretty good shape, because otherwise the stoneflies wouldn't be there. And so, yes, abundance can go both ways. A bigger driver or a bigger indicator would be species richness, which is the number of species. If you have a sample that's only got 5 to 10 species in it, there's probably an issue there where a normal healthy stream might have 30 to 40 different species.
And when you're sampling it out, figuring it out, are you usually focused just on a stream or a runoff or the whole watershed or rivers, which I assume are a lot bigger and harder to sample? And I know it's client-based, but how do you figure out the best way to sample to figure out what you're looking for?
Sometimes we know there's a problem already, and it may be a point source. And so that's a pretty straightforward thing where if you know there's a particular point source discharge, you can do upstream and downstream sampling and compare the two. And that's the method that actually federal EPA has a very specific methodology. And they use that kind of a method where they have an upstream or an unimpacted reference condition, and you base that against other samples that you know are in an affected area. Otherwise, a lot of these sampling techniques are just standalone sampling techniques, especially the Maine technique. You can pick a spot anywhere in a stream or a river. You can put in your samplers and get a result. And that's usually the kind of work that we end up doing. And I don't know if I would call it watershed based. I would call it maybe project area based, where you have an ongoing development, say a wind farm development, and there are multiple streams in the development area and road crossings and things like that. And so you would just pick sites in all the streams within that area, above and below culverts, and then you would take your samples accordingly. So it can go both ways. It can be a very specific type of sampling and analysis, again, for like a point source condition or a Superfund site, something like that. Or it can be a more general watershed sort of thing. And there's always usually other analyses that are going on along with that. It's never usually just a macroinvertebrate sample. There's often chemistry that's being analyzed for the water and the sediments. There could be erosion mapping going on. There's usually kind of a comprehensive ecosystem analysis.
And how do you sample?
Well, there's different methods. The easiest method is the one the EPA uses. So if you imagine a net, almost like a butterfly net, but sturdier with a heavier mesh and a flat bottom, so it rests flat on the bottom of the stream. You hold the handle up in the air and you press the net down into the bottom of the stream, and you stand upstream of the net and you disturb the substrate with your feet so that the organisms that are clinging onto the rocks in the sediment, they release from the substrate and the current carries them into the net. That's called a kick sample. That's the simplest way to do it. The method that Maine DEP uses is they use what's called an artificial substrate. So they take wire baskets filled with rocks, wire basket about the size of a loaf of bread, and you fill it with rocks, set it on the stream bottom, and leave it in there for 28 days. And then at the end of 28 days, you go back and you collect the entire basket with all the rocks, and you very carefully wash the rocks and the basket to get all the organisms out. So it'll just colonize. That rock basket will be colonized to just like the stream bottom will be colonized. And there are other methods of artificial substrates too. But that method, DEP likes that. Whenever we do a sample, we put in 3 of those. They're considered 3 replicates of one sample. And that's one sample location. They're very different, and they tend to work in different ways because the basket's filled with rocks. It tends to sort of pre-select for organisms that like to live in the rocks. Now, over time, since it's in there for almost a month, it will capture sediment in sand and along with leaves and sticks and whatever else is flowing downstream. So you will get a fairly good representation of the community of that stream. Whereas the kick sample, you're collecting what is right there, but you have to be careful because if you have multiple substrates in the stream, then you want to make sure you're sampling in all the different substrates. So you want to go from the sediment in the pools to the rocky riffles. You want to make sure you're collecting all those different habitats with the kick sample. But those are the 2 sort of methods that are usually used. For lakes, they have what they call grab samplers, which basically look like a little scoop, like a little dredge, and you drop it out of a boat into the sediment, and then you drop a weight down the rope, and it triggers a mechanism that closes the doors on the bottom of the scoop. And then you pull it up, and you've got a little scoop full of mud from the bottom of the lake. And so those are what they call grab samplers or dredge samplers.
Do you have any preference? Would you rather stand in the middle of a stream, or would you rather go out on a lake, or you just like it all?
Oh, it's all good. If I'm outside, yeah, it's all good. Rain, snow, it doesn't matter. A bad day in the field's better than a good day in the lab.
Wow, that is a great statement to make. I would imagine the time of year would have an impact. And I'm thinking, like hiking in Maine in the spring, if we have what used to be in the past, a normal snow year, there'd be a lot of snow melt and be really different than hiking and crossing a stream in August. So do you have to take that into account when you're looking at different species as well?
Well, if we were looking just for different species, yes. But the thing is most of the protocols that we are required to follow have specific sampling seasons. So Maine, for instance, you can't place your rock baskets until July 1st, and they all have to be retrieved by September 30th. And the reason they're doing that is that's actually the time of year where the insects, the life stages that are in the water, are under maximum stress because the temperature is usually the highest and the flow is the most variable. So that's when we do our sampling for Maine. If I were as a taxonomist, as someone who enjoys doing taxonomy and wants to have good specimens to look at, I would prefer to sample in March or April, which is more difficult logistically. But the reason is at that point, most of the insects, they're getting ready to emerge as adults. And the immature stage, it's when they're the largest and they have all of the characters well developed that I need to make an accurate species identification. They're big, beautiful specimens. If you do a lot of collecting in the late summer, you find a lot of specimens that essentially just hatched from the egg, and they're tiny and they don't have a lot of the characteristics. They don't have gills. They don't have wing pads. They don't have a lot of coloration, and it's hard to get accurate identifications. And we've actually done experiments with that just out of curiosity, we once placed rock bags in a stream in Unity every month for a whole year. So we just did January to December, and just to compare all of the metrics that are used, species richness, indicator species, different functional feeding groups. And we got very similar water quality results throughout the whole year. But it was a lot more interesting and a lot more fun to do the identifications on the late spring or late winter, early spring specimens. It wasn't a lot of fun collecting them in the middle, having to chop through the ice to get to your rock baskets in February. It wasn't a lot of fun. But the samples were really good and the specimens were beautiful.
Well, I'll go back to, you said a bad day in the field is still a better day in the lab.
There are some caveats with that, but yes.
Sure. So I guess that's one of the big differences in a way, is that the work that you do primarily is on water quality. So if you were just looking for the critters, then the sampling that you do would be that different type, right?
Right. Like, say I was doing a taxonomic review of a particular group of organisms, or a systematic review where I wanted to revise the systematics of a group and review the species descriptions. Then I would be going after the times of year when I knew I could get late instar juveniles or even adults. I would be going after certain life stages in a certain condition. Whereas for water quality issues, we need to follow the protocols that have been established for the relevant agencies. So if I don't follow the protocols for Maine DEP, they're not going to accept my data. So I've got to do it the way they laid out. And it's a good system. Like I said, DEP has been doing it for a long time. They've got a good system. They have a good model. The more data they collect, the more robust the model becomes, I think. And so it's a really good program.
Well, it's interesting that you said the water quality was consistent. So that seems to me an indicator that they have figured it out sampling wise. So the animals that you're looking for, the insects, I assume they're not microscopic, that you're looking for ones that you can see with the naked eye. Can you describe a few of the ones that people might see when they're out and about and tromping around in streams?
Sure. Well, while most of the invertebrates that we will find in streams, especially, will be insects, there are also mollusks and crustaceans and worms, and we identify all those as well. We call them macroinvertebrates because they are visible with the naked eye, but most of our work involves microscopes because we need to see fine structures on these things, fine details, especially to go to species identifications. So almost all the identification work is done with a microscope. And some really obvious ones that everybody has seen are water striders and whirligig beetles, which are shiny black beetles which spin around in circles on the surface of the water. Now, everybody has seen a lot of adult stages of aquatic insects, and they just may not know they're aquatic. Now, everybody has seen dragonflies. The nymphs of the dragonflies, the immature stages, are aquatic. They live in the water and then emerge to become the beautiful dragonflies that fly around in our yard, eat all the mosquitoes. But there are a lot of species that people wouldn't recognize as aquatic. So caddisflies, for instance, are very closely related to moths and butterflies, and the adults look very much like moths. If you live anywhere in Maine and you've left your porch light on at night in the summertime, there have been caddisflies at your porch light. There have been various species of midges at your porch light, which are small flies that look a lot like mosquitoes. And then, of course, everyone's favorite aquatic insect is the black fly. We all know that. Black flies are, of course, indicators of good water quality. When you have black fly larvae in a stream, it means that the water quality there's plenty of oxygen in that stream. The temperature is well regulated. So they are indicators for the most part of good water quality. And if you started turning over rocks in a stream, you would see a lot of mayfly nymphs. If the water quality is in pretty good shape, you would most likely see stonefly nymphs, and you would see a lot of caddisflies. And caddisfly larvae are some of my favorites because as they are related to moths and butterflies, the larvae spin silk, just like caterpillars of terrestrial moths and butterflies do. And a lot of caddisflies use that silk to make little structures. They'll either make a small case that they carry around, so the abdomen will be in the case and their legs will be poking out the front, and they'll carry the case around. As they grow, they enlarge the case or they cast it off and make a bigger case. and they make it out of sand grains or twigs or bits of leaves or pine needles. And then other caddisflies make what they call a retreat, where they'll actually build like a little tent or a little house attached to a rock or between a couple of rocks on the stream bottom, and they'll come and go from their little house. They'll leave the house and go forage on the stream bottom, and then go back in the house during the day to avoid predation from fish. And they'll actually build nets that act as filtering devices to collect food. So they build a net between two stones, and it collects organic matter, and they wander around and they clean the organic matter off the net, and then they can hide out in it to avoid predation. So if you looked around closely on the stream bottom, and you don't need any kind of equipment for this, you can just start picking up rocks and turning them over, and you'll see all sorts of things scurrying around. There's nothing that's going to hurt you. So you can just scoop up a rock and take some of the water and wash the water into your hands, and you'll see all sorts of things crawling around in there. They're incredibly abundant aquatic insects and invertebrates.
How long do they stay, the caddisflies? How long are they aquatic?
So it depends. Most caddisflies in this part of the world, they have one generation per year. So the adults will emerge in the spring, early summer. They'll reproduce. The females will lay eggs. Sometimes they lay eggs right on the surface. Sometimes they actually land on the surface of the water and swim down under the water and lay eggs on the substrate. The eggs hatch, and then usually the larvae are in the stream until the next summer. So they're in there for a full year almost. They molt 3 times. There's 4 stages of the larvae. So they're growing and they're molting while they're in the stream. And then just like a monarch caterpillar forms a little chrysalis and pupates in there before emerging as an adult, caddisfly does the same thing. It'll form a pupae in a little case in the water, and then emerge as the adult, swim to the surface, break through the surface tension, and fly off. There are other groups, mosquitoes, midges, which I mentioned before, they have a much faster life cycle. So they may have multiple generations per year. Some species have 2 generations per year, where they have sort of a short generation in the summer. So adults may lay eggs in the late spring or early summer. Those larvae will emerge before the end of the summer, but then their offspring will spend the whole winter and emerge the next spring. Dragonflies sometimes will spend multiple years as a nymph in the water, sometimes 2 or 3 years. Dragonflies are fairly long-lived as far as insects go.
What determines how long they stay in the water as nymphs? You said sometimes. Is it just different species of dragonflies?
Yeah, right. I'm referring to different species. Yeah, there wouldn't be, I don't think, a whole lot of variation within a species in terms of that. That's temperature dependent. They have what they call degree days. So a nymph or a larva has to have so many days at a certain temperature in order to fully develop to then pupate and emerge as an adult. But there's all kinds, when you start looking through the insects as a whole, there's a lot of interesting variations on that. There are species of stonefly that emerge in the winter. And if you go out alongside a stream, usually March, on a sunny day, you'll very often see stoneflies crawling on the snow alongside the stream. And they're small, black, they're maybe a centimeter or so long. Once you know how to spot them, you'll find them everywhere when they emerge, because they emerge in fairly high numbers. There's a really interesting species of mayfly that is named after a stream in eastern Maine. There's a stream down there called Toma Stream, and there's a Toma mayfly, which lives in floodplains, in sedge habitats and floodplains. And so they live in the stream over the winter. They're vegetarians, they're herbivores, they eat algae and things like that. And then in the last stage of the larval development, the snow melts and the sedge plains flood. And the mayflies move out of the stream channel into the floodplain where the sedge is, and they become predators, and they start feeding on other insects, mostly other mayfly nymphs. And then they emerge in the spring, and they lay their eggs, and their eggs don't hatch until early winter. In the summertime, you'll never find them. You can't collect the nymphs in the summer. You can only collect the adults for about a week when they emerge in the spring. So if you want to try and collect these nymphs, you've got to go out either in the winter or when the snow melts and the floodplains are flooded, and you can go into the sedge and collect. So they're very specific to that habitat.
Earlier, when you were talking about the different sampling and stuff, I was going to ask if you have ever come across a species that seemed new to you. And then I thought, well, that's kind of a silly question. But now that you mention this, and this one specific habitat of fly, do you feel like aquatic taxonomists have figured out all the different ones that are out there, or is there still kind of an unknown?
I don't know. I think most of the species are probably accounted for in terms of aquatic insects. Most of the work that we do, and I'm speaking we collectively as aquatic entomologists in this field anyway, most of the work that's being done is either practical applied work, like what I'm doing to make environmental assessments, or learning more about the tolerances and the habits of these organisms so that they can be used for that reason. The people who work on the evolutionary relationships and the systematics are constantly tweaking things. And especially we talked about molecular biology and genetics. That's been a really important tool in determining evolutionary relationships. So I always have to be up to date on the taxonomy, or else a name will change. Something will get moved from one genus into another genus, and I have to be aware of that. So there's that kind of research that's always going on. But I found, it's funny you mentioned that, because I was working on a sample about a month ago, and I found a species of stonefly in a sample that I've never seen before. And it's one of those species, I see it in the identification books and I've seen it in the key, and I'd always move right past it because I've never found it. And there it was. After almost 30 years of doing this, I have a specimen in my reference collection that I've never seen before. So that was pretty exciting.
I was going to say that must be really cool to feel like you finally got to see this thing that you hadn't ever seen.
It was. It's those moments, the longer you do this, of course, those moments become more rare because you see more and more. And a lot of the work I end up doing, I don't call it identifying, I call it counting, because I can look at a typical sample and I recognize almost all the species that are in there without using any reference material or any reference literature. And then I'm just counting how many of each species. So it is a lot of fun when I find things that are unusual or uncommon. And they're out there, so that does happen.
How long did it take before you did the transition from identifying to counting?
Well, it took a while. It was at least a good 10 years. And part of the reason is a lot of times for efficiency's sake in the laboratory, we divide up tasks based on who's better at different taxonomic groups. So someone might get all of the mayflies and someone else might get all of the caddisflies. And so as you learn more, then you start to branch out. And so then you have a new group to learn. And then there are other groups that require specific techniques, especially the smaller things. We talked about midges, which are these tiny little flies. Well, the larvae are equally tiny. They actually have to be prepared a particular way and mounted on microscope slides and looked at higher magnification for us to do the identifications. So when you embark upon learning a group like that, plus there's hundreds of species in that group. So that's a process that takes a lot of time. And so as I've gone on, I've branched out into different groups. And one of the other things we do in the lab is instead of just doing freshwater, we also do estuarine habitats and we do intertidal habitats. And so in the estuary, you will find insects. There are certain species of insects that are tolerant of salt water to a point, but then you start getting into more marine organisms. So there's the blend in the estuary. And then, of course, when you get into the intertidal, that's a completely different community. So no insects, much higher diversity of crustaceans and mollusks. And then all the other weird little things like tunicates and sponges and that sort of thing. So that's been a lot of fun because that's been a process that I've been working on for the last probably 15 years, is moving into more estuarine and intertidal habitats.
Well, that leads into what will probably be my last big question. How much do you see climate change and temperature? It's twofold, actually. Temperature increases in the water. This is, I would guess, one of these areas where even a small increase is going to have a relatively big impact on the animals. But then also there's a likely increase in the coming years, if it hasn't already happened in some places, of either salt marshes or more salt water coming into freshwater areas. How much are you seeing of that?
I would say not much, but I haven't been specifically looking for that. The kind of work we've been doing in the estuaries in the intertidal areas are very much like the work that we do in the freshwater where we have a point source problem. Almost all of those problems have been tank farms leaking or historical chemical contamination or that sort of thing. So we're looking at very specific things. In terms of temperature overall, there is some data showing that there's a kind of a northward shift in certain species, particularly in smaller, higher elevation streams, places like the White Mountains. And it makes sense in a way because those streams are, in a way, I don't want to call them pristine because there's always input from pollution and rain or whatever, but they're probably as close to being pristine streams as we can get. And so the organisms that are living in there are already organisms that are generally intolerant of things like pollution. And what's happening is you're getting not just an increase in the temperature overall, but you're getting much higher variability in flow in those streams. With the more frequent and heavier rain events, it's adding to the stress in those streams. Larger water bodies, freshwater anyway, seem to be coping with it a little bit better. Like, I haven't seen a lot of change in the overall community structure. I went back and I looked through historical data. The one nice thing about Maine DEP having this long-term water quality modeling thing is the data is all public information, and they do have specific site location that they've sampled on almost a yearly basis for the last 20 or 25 years. And so I went back and I looked at data from some of their long-term stations to see if I could find any major changes in community structure or just temperature, and didn't really see too much. But those systems are variable to begin with. And you can collect in the Kennebec River one year and go back the next year, and the flow will be 15 times higher than it was when you were there the year before. And so it's hard to control all those variables. With the intertidal stuff, again, you're seeing the more frequent storms and storm surges, which cause problems with the substrate. They're also causing the flooding, which are washing more contaminants into the water. But then you have the added temperature change and the pH differences that are really affecting the mollusks in the marine and the intertidal communities. And then, of course, the invasive species. I can think of, there's an invasive snail that's a freshwater snail. Most of the freshwater species you hear about being invasive are fish. You don't hear a lot about invasive caddisflies or whatever, mosquitoes occasionally that are not native. But the invasive species in the marine and the intertidal habitats are serious and are causing problems. You know, the green crab is the one that comes up a lot as being a real problem species. I keep hearing about blue crab, moving further and further north, and that's just going to keep happening with other marine species. And I think it's going to happen with the aquatic insects too. You'll see a northward shift. Back to black flies larvae not only require well oxygenated water, but they like fairly cool water. That's why you find them more in the shady streams in the woods. You tend not to find them in a more exposed area where there's a lot of sun. They just don't like that. So you'll see probably a shift in the black fly community.
Thanks for that. At the risk of making you pick a favorite child, do you have a project or two in particular that you just found really cool and exciting that you worked on?
I did. This was a project that I did twice, and this was a USGS project. And this wasn't actually larval forms of aquatic insects. It was involving adult forms. But there was a researcher in Wisconsin for USGS who was studying the bioaccumulation of heavy metals in birds. And this was in relation to, I think, there was a lead mine in Colorado. And the reason I'm hesitant about this is because I don't always get all the details of where these samples come from. But what she was doing was she was collecting what she was calling a bolus. So we're going to have to get into birds now. So a tree swallow will fly around and eat insects, catch insects, and it doesn't digest them. It stores them in the crop. Well, birds have a crop, which is the storage organ before the stomach. And then it regurgitates this up for its young in the nest. So she had a method of getting into the nest and collecting the bolus before the chicks could eat it. And the reason she was doing this was she wanted to know if they were collecting aquatic insects, because that's where the contamination was. If they were only eating terrestrial insects, they weren't going to be useful, because she wanted to go on and collect blood samples from the birds and look for the lead. Well, if they're eating terrestrial insects that aren't in the sediment, they're not going to have the lead. So I would get vials of, they looked like little, I don't even know what they looked like, little balls. It almost looked like a pea. And I would put it under the microscope and I would gently unfold it. And it would be this mashed up ball of insects. But they were all fully intact, all the legs, all the wings, just compacted into a ball in the crop of a tree swallow. And so I would gradually unfold all these things. And then I would go through them and identify them and determine which ones were aquatic and which ones were terrestrial. I did that 2 projects. She sent me probably 100 samples. We called it the Bird Barf Project. It was amazing because I learned that tree swallows will eat anything that flies, no matter what it tastes like or whether it has a stinger on it or not, it'll go down the hatch. And sometimes I would have a bolus that would be a single insect. It might be a damselfly or a paper wasp crushed into this little ball. Sometimes I would take the little bolus apart and there would be 35 mosquitoes in it or 40 little midges. Sometimes there'd be 30 insects in there and each one would be a different species. It was almost like getting this little Christmas present and just unwrapping it to see what was inside of it. It was a fascinating project.
That sounds like the best lab work you could do because you don't know what's coming.
You don't. Yeah, it really was. It was a lot of fun. It was challenging too, because while initially when I started learning about insects and becoming an entomologist and training, you do a lot of adult work. You do a lot of adult identification. It's a whole different set of characters and morphology that you have to learn. But then when you get into the water quality stuff, you're dealing mostly with the nymphs and the larvae. And so I had to do a lot of literature research and obtain the identification keys in the literature to do the identifications of the adult forms. And usually I could tell right away if it's an aquatic, if it's a mayfly or a caddisfly, I know that. But she wanted species if we could get there. And it was a challenge. It was a lot of fun.
How long ago was that? I'm just curious.
The last time I did that was probably 12 years ago. 2 systems, one river in Colorado, and I think one was in Wisconsin. That was the lake, maybe Minnesota, somewhere in the upper Midwest. There was a copper mine, and they were doing this processing, and they were ending up somehow with sand as a byproduct, and they were dumping it in a lake. And it was contaminated with copper, and they were just dumping it into this lake. And you can walk into this lake now, and you're walking on the sand, and the sand is like 4 feet thick, and it's all this copper contaminated sand from the lead. And so the insects are just doing what they do. They're burrowing into the sand and eating detritus and doing whatever, and then getting loaded with copper. And then in the Colorado project, it was lead.
Yeah, 4 feet is a lot.
Yeah, they processed copper for probably 50 years from that mine.
Right.
And just, yeah.
That's a pretty cool project too, because it has to bring together all these different pieces of science to figure out what's going on.
Yeah, and there's been other interesting, there's a lot of small stuff that goes on. A lot of it's just general habitat quality. It is always interesting when you have something specific in mind. It has a level of importance, I think, to it at that point that isn't always present when you have a super fun site. We did work on the Housatonic River, which is historically heavily contaminated with PCBs. And they've done experiments with dredging the Housatonic to remove the contaminated sediments. And so we would do identifications of the macroinvertebrate community before and after the dredging to see what the results were, whether or not that's an effective technique for removing the contaminated sediment. And it is, in case you're wondering, it's phenomenally effective. So I think we had 5 to 10 species pre-dredge and 40 or 45 species post-dredge after the river had a chance to kind of recover its normal substrate after the dredging. So that kind of work's always really interesting when you can see very specifically in the resident community whether or not something has been effective or not.
Well, John, this has been just a delight to talk to you. The best thing for me about this little side project of mine is that I get to learn about all this really cool work from people who are super enthusiastic about it. And I feel like just a little bit smarter every time. So I really appreciate it.
Oh, well, thank you for having me. This has been fun. So I don't know, maybe we have to get a nice little collection for you for the museum.
Oh, that would be neat. If you can send me some pictures of the rock sampling basket, actually, that would be really cool. We're actually in the middle of a revamping and redoing a river exhibit, so we could figure out a way to incorporate that in there.
Oh, yeah, that'd be great. And somewhere on a thumb drive, I have bird barf pictures. So I took a couple photos of the complete bolus. And then once it had been separated into its component bug parts, like a before and after picture.
We can't turn that down. You're going to have to find that for us. Thanks a million, John. I really appreciate it.
Well, thank you.
You're welcome. Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. The Discover Maine theme was composed and performed by Nick Parker.